Hello everyone. So instead of just talking through a Zoom camera for the semester, I feel it's going to be easier for me to present the ideas at these sections by doing small little YouTube videos. So we're going to start these off today with how gametes are formed. In these videos, I will give you the information you need that you want to be looking at while you're going through this. So this is section 2.7 of the book. The figure you're going to be looking at while we're talking about this is figure 2.17. So this shows us both spermatogenesis and oogenesis. So this is going to be a lecture, a small video about these two things. You also may want to have your assignment up while you're reviewing these, because you'll be able to see the terms that I point them out as I point them out. Maybe you might want to put a check mark or highlight them as you watch. And then I will go through and I will talk about different things. Spermatogenesis and oogenesis are going to allow us to see how mitosis and meiosis apply to the creation of gametes. In humans, the gametes that we're going to be concerned with are the sperm and the egg. But to get to a sperm, you can see it's actually quite a long process. If you look at figure or table 2.6, this entire process is going to take us about 64 days. A lot of people think sperm are easily created. They are not. It is about a 2-month process from when the spermatogonia actually begins mitosis till when we actually get a viable sperm cell. So, in the testes is where we find the spermatogonia in males. These are in the scrotal sac. They lie beneath the penis. These are specialized cells within the testes that will undergo mitosis in order to create sperm. So the specialized cell that will create sperm are called spermatogonia. As you can see, they are diploid. They will undergo mitosis for the entire length of a male's life. As they undergo mitosis, they will then produce primary spermatocytes. These are the first cells that will undergo meiosis. So you can see the primary spermatocyte is also a diploid organism or diploid cell. The role of meiosis 1 is to separate those homologous chromosomes. So we can see the primary spermatocyte undergoes meiosis 1, and this can create 2 secondary spermatocytes, and you can now see that we have divided the genetic material in half. These cells are haploid. N will always represent haploid. These cells then undergo meiosis 2. This is when we will now separate the sister chromatids held together by that centromere, and we will then have 1 chromosome for each in our final product, and we will have 4 spermatids. Notice this is not a full sperm at this point. This is just a cell. It will take another 2 weeks or so as that spermatid matures. It will then become a viable sperm, and it will now have the genetic material in its head, and it will have a tail or a flagella. This is one of the only animal cells that typically has a flagella. And it's not just in animals as well. Most sperm in plants, both higher and lower plants, also have flagellated sperm. This was one of the evolutionary steps in plants that allowed higher plants to come about. I know that in the past this class has been taught mostly in terms of animals, specifically humans. I will also bring in other caveats. So that is spermatogenesis.